Welcome back, everyone. Well, I'd like to start off by thanking once again ISS National Labs as today's sponsor for the programs we're enjoying all day. Please stay tuned for a human research panel monitored by Jennifer Buckley at 2:30. But now we turn to Ken Shields, who will moderate our next panel on the ISS National Labs. Ken Shields is the Vice President and Chief Operating Officer at the Center for Advancement of Science in Space, the managers of the ISS US National Laboratory. Ken first joined CASIS in 2012, and during his tenure has directed the Flight Operations Department, served as the NASA liaison, and led the CASIS STEM Education Department. In 2017, Ken was awarded NASA's Outstanding Public Leadership Medal, and over the past 3 years, he served as a member of the ASGSR board. In his current role as COO for CASIS, Ken oversees all aspects of the organization's business operations as his team seeks to advance our nation's leadership in commercial space, pursue groundbreaking R&D not possible on Earth, and leverage the International Space Station to inspire the next generation. Ken?
Thank you, Doug. That was a very nice introduction, and I'd like to welcome everyone to the ASGSR plenary panel entitled ISS 2.0 for 2020. I want to first start out by saying thank you. I want to express our appreciation to Doug and the entire ASGSR team and really the entire society for giving us the opportunity today to share a little bit with the audience here about what we are doing today and in the future at the National Lab, and also hear from a few of our current researchers, current and future researchers, who will be conducting microgravity research in the not too distant future, and many who have conducted some here in just very recent years. So we're looking forward to a great panel today. I also want to thank our audience for tuning in. These virtual conferences, I can't tell you how important they've been for the entire community, and everyone will have an opportunity to participate in today's discussion. If you have some comments, questions, please, during the session, go ahead and chat those out, and at the end of all the presentations, we will allow plenty of time for conversation, discussion, some Q&A, and address any comments, questions, concerns that you didn't hear during the discussion or the panelist presentation. Today's session is going to feature a diverse set of panelists. I'm really excited about this conversation. We're going to cover, I think, a lot of aspects of what we're doing on Space Station today from a National Lab perspective. First, and in just a few minutes, we'll be joined by Mr. Alex McDonald, who is the NASA chief economist and also the NASA program executive for the ISS National Lab. Alex and I will have a brief discussion about where we have been from a National Lab perspective, how NASA views the National Lab moving forward, what our current status is, and we'll even get into what some near-term initiatives are that we're working through to set the foundation for a new vision for the future of the National Lab. After that discussion, we'll segue into some panel presentations from researchers. We have Drs. Se-Jin Lee from the Jackson Laboratory, Nicole Wagner from Lambda Vision Incorporated, and Dr. William Wagner from the McGowan Institute of Regenerative Medicine. And they will be coming up also later after the discussion. They've got some presentations, and as I said before, we'll get into some discussion after that. So as a brief intro, I think most of this society is well aware of the past 20 years, and oh, by the way, we just celebrated a big 20-year anniversary on ISS, 20 years of human habitation. I know that we're all excited about that, and I know here at National Lab in CASIS, we've been talking about it for weeks and weeks. But I think everyone's well aware of how valuable the ISS has represented numerous communities, certainly in the early days, learning how to live, work, and explore space. In addition to that initial and primary mission, the ISS has evolved into this amazing multi-purpose, multi-user facility where literally thousands of investigations are going on or have taken place over the years in every discipline that you can think of and from just about every organization type that you can think of. So over the years, it's really transitioned to this terrific public enterprise that has now been opened up for non-NASA use and new to space users to explore other avenues of research and technology that may not be directly related to space exploration. We're investigating things such as new product development, new technology development, once again, in just about every application that you can think of. In the early days, when CASIS first took up the charge of managing the ISS National Laboratory, and we do that in partnership and in collaboration with NASA via a cooperative agreement. Really, the charge and the mission at that time was to break barriers to new to space users, introduce non-NASA users and different companies and organizations that really hadn't contemplated or thought about what microgravity or the extreme environment of space or even the vantage point from LEO could do for their research, whether that be fundamental science or applied research and development or technology maturation, technology development, and technology demonstration. And now, all those years later, after 8+ years of lowering those barriers and forging important relationships and collaborations with amazing government agencies like National Institutes of Health and National Science Foundation, Department of Defense, Department of Energy, and many more. In addition to industry partners, we now have some really important history and data that are shaping what the future of the National Lab will be. Initially, it was to break down barriers, achieve full utilization, bring new interest and new partners to space to do research with us. We've achieved just about 100% utilization. We have created several new public-private partnerships to expand our capabilities in space, these commercial service providers who are microgravity research and technology experts. And now it's time to evolve and turn our pursuits towards a more directed and thematic programmatic research effort at the National Lab. We're in the midst, or really the very beginning, of starting to go down that pathway now. And so I think now perhaps might be a good time for us to have Alex join the conversation. Alex, welcome. I'd like for you to now join in some conversation with me, and we can talk about how you recently joined. I think it was in about the April 2020 timeframe. NASA chief economist, as I said before, and now you've got the great honor and responsibility of taking on the National Lab as the NASA program executive, and we're just really happy to be working with you. I wonder if maybe initially you could share what some of your initial observations are, insights, and let's talk about perspectives and what the future might bring. Welcome.
Yeah, well, thanks, Ken, and appreciate the kind introduction, and great to be back with the ASGSR community. I actually think the last ASGSR meeting I attended was, I think, in 2011 in San Jose. So good to be back, if only virtually, and of course, good to be talking with lots of friends and colleagues here once again. I think Ken gave a really great overview of where we are with the National Lab. As I suspect folks are aware, there was an independent review team that was commissioned by Administrator Bridenstine last year, the report of which came out earlier this year that it had some findings and observations that were both critical of NASA and CASIS management. But the report also found that the utilization of the space station for new economic activity, for fundamental science, has really been proven out. And so we've taken on board, the lessons from the report, but also the encouraging findings, and really recognize at NASA that the principle of having the space station and half of its resources for research used for non-NASA research as a national lab is a fundamentally valuable mechanism for encouraging new research across the United States of America. And one of the shifts that I think is important is really this recognition that it's a national lab and a public service and utility, right? Those of you who have been familiar with the last 10 years or so knows that at the beginning there was a little bit more kind of question of maybe the National Lab was itself going to be type of self-supporting economic enterprise and this kind of stuff. And in fact, what we found is that actually where the National Lab works best is when it is this public service around which dozens of other companies then build their own self-sustaining businesses. And that's in fact what we've seen very successfully. So we've taken that on board and really focused in on some of these vital national interests that the National Lab can serve. And a couple that I want to highlight for this community, because I think you are all are critical partners in the success of these enterprises in particular. And one of those is, of course, fundamental science, and specifically fundamental science as supported by our interagency and other government agency partners. When I came on board as program executive in April, one of the most interesting, encouraging signs that I found that I wasn't fully aware of was really how much the NIH and the NSF have been using the National Lab to conduct their own program activities. Over $40 million combined between those two organizations spent on research to use the space station. That's a really impressive metric as an economist, because it means that they find that fundamental value. And that's really an area that we want to see grow. I think that's something that, if you're out there and, you've got contacts out in those agencies, right, it would be great to figure out how do we as a collective national government industry community figure out how to maximally utilize the space station for these programmatic endeavors that our interagency partners want to use. So I think that's something that we've seen great success on in the past. We want to build on that success and really make a significant focus going forward. The other that I wanted to highlight as well, because this is certainly a priority of the program for quite some time, and also a priority for the administrator, is really figuring out how we can produce something in space utilizing principles of microgravity. I'm sure everyone in this community is familiar with both all the different efforts that have been undertaken over the decades, going back to the space shuttle in that area, and also how very, very hard it is to do that. Right? No one is under any illusions that this is an easy process. At the same time, I think there is a recognition that it's a fundamentally important goal for the National Lab and for all of us to really work towards, because whether it's something that's produced and sold on Earth, or whether it's a scientific process that is so valuable that it's done again and again and again, this is going to be something that is going to be able to change minds if we can achieve it. And what's exciting to me is that actually the program entity within NASA that has the responsibility for achieving that is the National Lab, right? The National Lab program that is conducted in cooperation with the operating partner of the National Lab, CASIS. And so for me, it's been really exciting to see the team at CASIS kind of take on board this new challenge. I'm sure you're aware there's lots of other different funding streams related to this in SBIR, under the ISS NRA. And that's going to evolve in the months and years to come here, but I think it's just important for us all to know that this is an important goal for NASA. It's an important goal for the National Lab, and it's going to be a hard one and an exciting one, and we're going to need a lot of support in the biotech, bioprinting, and in the material science areas, and all these different technological areas that could potentially kind of get that milestone, right, of a thing that is first made in space and sold on an annual basis on Earth. That will be a historic NASA and space community first, and it's exciting to be working with everyone here to try and make that happen.
That's great. Thank you for that, Alex. And wonder if we might share with the society, as you mentioned, the ASGSR and its members. They're really important community members, if you will. Really important society. And a feedback group, and also a user community for ISS National Laboratory. And we've got 2 or 3 important initiatives that we're working on right now. One of those relates to exactly that, which is building out user community, building out more formalized and institutionalized structures so that we can get good feedback and get good advice. And one of the things that we've embarked on, and I think many folks out in the audience, certainly members of the society, know that we're in the midst right now of standing up an ISS National Laboratory User Advisory Committee. And the exciting news is, I think here in just the next few weeks, we will be in a position to announce exactly what that membership looks like, what perhaps the first meetings may address, and have some very public information about what that might look like. Alex, you want to talk a little bit about the User Advisory Committee and its role, and how important it's going to be in informing what the future of the National Lab will be as far as strategic objectives, priorities, and even prioritization of resources and where we use them.
Yeah, no, that's great, Ken, and I'll try to make it quick because we could talk about that forever, but we want to get to our panelists here who've got the actual really exciting information, which is all the exciting actual science technological developments. User advisory committees are a really important new mechanism to really ensure that all of you who use the space station for your research are able to provide advice into the executive leadership at CASIS, the executive director, to essentially establish some of these priorities under what are essentially going to be kind of 5 program lines that are emerging. And the composition of the advisory committee is very similar to the NASA advisory committee. There'll be 5 subcommittees in each of these program areas: fundamental science, education, commercial service provider use, in-space production, and tech dev. And these committees are going to be really critical in establishing, the important technical areas and important priorities for the National Lab going forward. The team at CASIS and certainly NASA management team is certainly very much looking at user advisory committee to be a critical source of input and prioritization information, so that we can make sure that the management of the National Lab is really done according to the needs of the users. As we all know, users are very diverse, and it's a very challenging process to manage these portfolios. But that's all the more reason why we're going to have a public forum where these things can be heard and understood by the users. And frankly, I'm really looking forward to it, because I think we're going to be looking to that community for great insight into what the most promising areas for future research might be. And yeah, I think that's a great one going forward. And just one last piece before I think we turn it over and get into some of those exciting results. Another related area is going to be having a new proposal prioritization and selection evaluation processes that are going to align with these program areas, that are going to be regular calls. And that's another really important takeaway that we had from the independent review team. And I think that's going to be really something important for this community, because it'll start looking a lot more like the NASA proposal evaluation processes that I think you might be familiar with. And having those regular calls, I think, is going to help the community out. So a lot of exciting stuff, and yeah, just great to be working with the team to make it happen.
Well, thank you. Appreciate your conversation today, and maybe at the end of the panel presentations, we get some more participation from the audience. But from a CASIS perspective, I can tell you this renewed partnership and renewed focus moving forward with NASA has been great. And Alex, you've been a great addition, so we really appreciate you getting on board and taking us into the future. Appreciate it. So with that, we would like to now segue into our researcher panelists. It's my great pleasure to introduce our first presenter today, is Dr. Se-Jin Lee. Dr. Lee is from the Jackson Laboratory, and his primary research interest is to understand the role of signaling molecules in regulating embryonic development and adult tissue homeostasis. Much of Dr. Lee's work is focused on a molecule that they have designated as myostatin, and they have shown that myostatin is expressed specifically in developing adult skeletal muscle, and that mice engineered to lack myostatin exhibit dramatic increases in skeletal muscle mass throughout the body. Dr. Lee and his team are currently attempting to elucidate the mechanism of action of myostatin, as well as the mechanisms by which the activity of myostatin is regulated. Their long-term goal is to attempt to exploit the biological properties of myostatin to develop novel therapeutic strategies for treating patients with muscle degenerative and wasting conditions such as muscular dystrophy, sarcopenia, and diseases like cancer, AIDS, and even sepsis. So Dr. Lee, welcome. Thank you. And please carry on with your presentation. I'm sure today's audience has a lot of interest to understand what you did on the ISS, how microgravity research moved your research down the line.
Thanks, Ken. Well, today I'm going to tell you about a project that we just did where we sent mice to the ISS. And this is a story that begins about 30 years ago. I just started my own lab, and I got interested in this group of proteins that's referred to as the TGF-beta superfamily, as Ken mentioned. These are proteins that are all structurally related to one another, and they're made by a given cell in the body, and then either act locally, signaling to neighboring cells, or act as hormones by signaling to cells located somewhere else in the body. And I was interested in this group of proteins because these have been shown to be potent regulators of cell growth and cell differentiation, and so they had the potential for really a wide range of clinical applications, for example, with respect to tissue repair. Now, at the time that I got interested in these proteins, about a dozen members of this family were known. And it seemed likely to me that that was just the tip of the iceberg. And so we set out to search for new members of the family essentially by guessing as to what the amino acid sequences of new family members might look like. Well, this slide shows what the superfamily looks like now, and it comprises nearly 40 distinct genes. And the ones highlighted in red are ones that we identified in our screen, and we named these just generically GDFs for growth differentiation factors. And the focus of my lab ever since has really been to try to understand the biological functions of these molecules. Now, one of the approaches that we took was to generate mice in which we deleted these genes individually in the germline and then looked at the consequences of that. And when we did that for one of these that we originally called GDF8, we ended up with a rather dramatic phenotype. And so the mouse on the right here is a completely normal mouse, and the one on the left is a mouse that completely lacks this gene GDF8. And it turned out that these mice, the mice lacking GDF8, had dramatic increases in skeletal muscle mass throughout the body, with individual muscles growing to about twice the normal size. So this told us that GDF8 normally acts to limit muscle mass. And so we decided to rename this gene myostatin. Now, the original studies were done in mice, but since then now it's clear that the function of myostatin has been conserved in many other species as well. Here's an example of a naturally occurring mutation and the result of a naturally occurring mutation in cattle, and it results in this double muscling phenotype that had been identified by cattle breeders for many years. We also identified a myostatin inactivating mutation in a human, also in a very heavily muscled individual. So all of this immediately raised the possibility that if we could find a way to block myostatin activity, that we might be able to increase muscle mass in patients with muscle loss. And so, for example, the question was whether by blocking myostatin, we might be able to help patients with muscle degenerative diseases like muscular dystrophy, or people with muscle wasting due to diseases like cancer, AIDS, heart disease, kidney failure, and so forth. We're also wondering whether this might be a strategy to help combat the muscle loss that occurs during aging. And so the focus of our research for many years now has been to try to understand how myostatin works at the molecular level, at the cellular level, and physiologic level, really with the goal of trying to develop ways to modulate myostatin activity, again, with the long-term goal of trying to treat patients. Now, in addition to biochemical and in vitro studies working out the details of how myostatin works, we were also then testing ways of blocking myostatin in mouse models of human diseases. Now, one of the models that we were interested in really from the get-go was the possibility of testing this in the setting of microgravity. And so one motivation for this was that the question being whether this might be a strategy to help combat the muscle loss that occurs in astronauts during space travel. But in addition, microgravity was a great way to mimic the type of systemic muscle loss that you see in as a result of disuse. So for example, people who are bedridden or in wheelchairs or in the elderly. And so over many years, I tried to reach out to various people at NASA to try to get this project off the ground. At one point, I even decided to try to appeal directly to the NASA administrator, who at the time was Michael Griffin. And I decided to take advantage of the fact that over the years, these mice that we had generated lacking myostatin had received quite a bit of press coverage. So here's some examples of this. And you can see that whenever these mice were described in the popular press, they were referred to as mighty mice. So I sent this letter to Michael Griffin. You can see it was dated 2006, in which I explained very briefly what I was interested in doing. And so I laid out what the goal of the project would be. And I suggested that we could call this project Sending Mighty Mice to Space. Well, I never received a response back to this proposal, but the opportunity to do this project on the ISS came in the form of an email sent by, this is 12 years later, sent by Liz Warren at ISS, addressed to Ed Liu, who's the president of Jackson Laboratory, asking, and this, in the interim, I moved to the Jackson Laboratory, moved my research operation there. And the email was asking whether the Jackson Laboratory, with its heavy focus on mice, might have some projects that might be interesting to carry out at the ISS. And so Ed contacted me, and in about 3 milliseconds, I put together a proposal, sent it back, and it got approved last year. And so this project had two arms. One arm, a genetic arm, involved sending these mice, these mighty mice, lacking myostatin, to the ISS. And then the second arm was to send normal mice to the ISS, but then block myostatin activity pharmacologically. And for this purpose, we used a biologic based on one of the receptors for myostatin. So we had shown very early on that myostatin acts by binding to cell surface receptors. The initial binding is to what are called the type 2 receptors, ACVR2 and ACVR2B. This binding event then leads to engagement of the type 1 receptors. And then it's this complex that initiates all the signaling, the intracellular signaling that leads to inhibition of muscle growth. Well, our collaborators at the pharmaceutical company Wyeth had developed a truncated form of one of these receptors, ACVR2B, which contained the domain of the receptor capable of binding myostatin, but lacking the rest of the receptor. And this is a potent inhibitor of myostatin because it binds to myostatin and prevents it from seeing its true receptor. And for this reason, we refer to this as a decoy receptor. And this is a very potent, in fact, it's probably the most potent muscle anabolic agent that's been described. So we showed that you could give mice just 2 injections of this decoy receptor systemically and get the muscle to grow by over 50% in just 2 weeks. Now, this particular biologic, this decoy receptor, in addition to blocking myostatin, is also capable of blocking another protein related to myostatin called activin A. And activin A plays a similar role to myostatin, but in this case, with respect to bone. And so this decoy receptor, when you give it to mice, also has a pretty dramatic effect in terms of increasing bone density. So with a single biologic, we were able to look at the effects of blocking this pathway on both muscle and bone loss, which of course are 2 of the most important health concerns that astronauts face in microgravity. So here's the timeline for the project. So the mice were launched in December on SpaceX 19, and then they spent about a month at the ISS, and then splashdown in the Pacific Ocean occurred in January. All 40 mice that were in this project were delivered live to us in San Diego, where we had rented out a laboratory. And then some of the mice we analyzed right away, and then some of the mice remained at that laboratory for an additional 2 weeks in what we call the recovery phase of the project, where we looked at the effect of giving this biologic in terms of recovering muscle and bone mass. Well, I'm going to just, if I had to summarize the results of this project in just 3 words, I would say it basically worked, meaning that the mighty mice, the mice lacking myostatin that started out with about twice the muscle mass, retained most, if not all, of that muscle during the stint at the ISS. And the mice in which we gave this biologic, this decoy receptor, the decoy receptor had a very dramatic effect in terms of increasing both muscle mass and bone density in the setting of microgravity. So this was very effective on both muscle and bone, even though the mice were unable to bear weight. Now, in addition to analyzing the gross tissues, we also carried out a detailed molecular analysis of tissues. And one of the things that we looked at was the plasma profile of proteins. And so here we worked with a company called SomaLogic that had developed a platform whereby they could measure levels of thousands of individual secreted proteins from just a single plasma sample. And what I'm showing you here, if you just focus on the bottom heat map there, it's just showing graphically the top 50 upregulated proteins and the top 50 or the bottom 50 downregulated proteins in the plasma as a result of treatment with this decoy receptor. Now, this is in mice that were on the ground. And what was striking about this analysis was that amongst these proteins that changed were a number of proteins, components of key regulatory pathways involved in muscle and bone homeostasis. And so, for example, components of the TGF-beta pathway itself, the Indian hedgehog and sonic hedgehog that are very critical for bone maintenance, the Wnt regulatory system, the RAGE regulatory system, and then the RANK regulatory system. And so all of these have been implicated in muscle and bone homeostasis. And we showed, looking at the molecular analysis of RNA expression in muscle and bone, that many of these pathways were also clearly regulated in those tissues. Now, it turns out that many of these pathways were also seen as regulated in flight versus ground mice. So as a result of being in microgravity. And what was striking was that some of these pathways regulators were regulated in opposite directions, which is what you would expect given the opposite effects that you see as a result of microgravity versus this decoy receptor on muscle and bone loss. And one of the most exciting, tantalizing piece of data that came out of that was that myostatin itself seemed to be upregulated in the plasma in these flight mice, suggesting that myostatin itself might be involved in muscle loss that occurs in space. And of course, this was the entire motivation for our project from the get-go. So we're very interested in following up on a lot of these leads to try to better understand what these pathways are doing. And so that's certainly one focus moving forward. And then we're also interested in continuing to explore this type of therapeutic approach for clinical applications, both for people on Earth as well as perhaps for astronauts during space travel. And we're hoping that we'll have the opportunity to test some of these ideas in further experiments at the ISS in the future. Well, we recently just published these findings in the journal PNAS, and I just wanted to acknowledge Emily Germain Lee is a world's expert on bone biology and bone disease, and she was the co-investigator with me on this project. Emily also happens to be my wife. And then of course, none of this could be done without the extraordinary work of the 3 astronauts on board the ISS, Jessica Meir, Christina Cook, and Drew Morgan, who carried out all of the actual procedures aboard the ISS. Thanks for the opportunity to tell you about our work.
Thank you, Dr. Lee. That was a great presentation, and looking forward to having some more discussion in just a few minutes about your research. Before I move on and introduce our next panelist, I want to remind the audience, please, if you have any questions, comments, go into that chat box, go into that chat field, and please populate that so that we can include that in our discussion at the end of the panel session today. So our next panelist is Dr. Nicole Wagner. Nicole is the President and Chief Executive Officer at Lambda Vision. She obtained her PhD in molecular and cell biology from the University of Connecticut under the advisement of Dr. Robert Burge, who is a pioneer in the field of light-sensitive proteins. Nicole spent the majority of her graduate career working on optimizing retinal-containing proteins for application in devices and during the course of her PhD research. She played a critical role in the proof of concept experiments which helped to found Lambda Vision. Through the use of site-directed mutagenesis, site-specific saturation mutagenesis, and directed evolution, Nicole and her team were able to genetically engineer the protein bacteriorhodopsin.
You got it.
Thank you, Nicole, for a variety of device applications, including protein-based holographic and three-dimensional memories, a chemical detection sensor, and most recently, a protein-based artificial retina. Dr. Wagner, welcome, and I'm sure today's audience would appreciate learning about your current microgravity work and maybe even a little bit about the future plans at Lambda Vision.
Wonderful, Ken. Thanks a lot for having me. So I think you did a great job introducing what we're doing, but just as an overview, my name is Nicole Wagner, and what we are developing here is a protein-based artificial retina to treat patients that are blinded by end-stage retinal degenerative diseases. So the 2 most common are macular degeneration And then there's the orphan designation of retinitis pigmentosa, which many people are also very familiar with, that both lead to irreversible vision loss. So I like to start off my presentations, let's see here, by just sort of setting the scene. So I ask everybody to imagine being at the height of your career and being forced to give it all up because you can no longer see a computer screen. Imagine having to ask your friends and family for a car ride because you've lost your license. Now imagine finding out that you're about to be a grandparent for the first time, only to know that you'll never be able to read your grandchild a book. Age-related macular degeneration and retinitis pigmentosa, they are devastating diseases which ultimately lead to blindness. Retinitis pigmentosa, as I mentioned, is an orphan designation. It affects about 1.5 million people worldwide, and it tends to affect the peripheral vision first. So you start to lose the vision on the sides, and then that'll narrow until a patient is completely blind. We then plan to move on to the larger market of macular degeneration. That affects about 30 million people globally, and even you can estimate that there's more people than that. And this disease works in the opposite way of retinitis pigmentosa. So these patients lose their central vision first, and then over time that'll start to expand until a patient is until a patient is completely blind. Age-related macular degeneration is the leading cause of irreversible vision loss in people over 55. So it's affecting many, many people. Both of these diseases have no cure, though there are some treatments available. And so the current treatments that are available, they only slow the progression of the disease. And so what you'll see here on the right-hand side is the current standard of care. This is a technology put out by Second Sight. And what you'll notice is that this is very low digitalized resolution, barely enough to distinguish between light and dark. And this current technology has a reimbursement precedent of over $75,000 per eye. And so it's really just not good enough. And so patients are really needing a much more clearer picture and better resolution in order to be able to see. So what our solution is we have a small, simple, flexible implant which can be placed in the back of the eye through a simple surgical procedure similar to a retinal detachment procedure to provide high resolution vision for these patients. The implant is intended to be long lasting, and our goal here is really to replace the function of the damaged rods and cones of the eye. So these are your light sensing cells. And we do this by using the light activated protein bacteriorhodopsin. Bacteriorhodopsin is a 7-transmembrane alpha helical protein which pumps a proton from the intracellular surface to the extracellular surface of that protein. And it does this because it contains an all-trans retinal chromophore, which is covalently bound to a lysine residue in helix 7. It's that covalent binding which gives this particular protein some of its stability and makes it slightly different than the native signal carrier rhodopsin. The protein that we actually use is formed in this 2-dimensional hexagonal lattice of trimer, which is also known as the purple membrane. This is incredibly stable, and so bacteriorhodopsin in this particular form is stable to almost over 85 degrees Celsius. So it's an incredibly stable protein and an incredibly stable construct. It's that construct that we actually use to make our artificial retina. So we have an ion permeable membrane, we use a polycation, and then we use multiple layers of this protein, polycation, until we build up a sufficient number of layers that we can absorb enough incident light to generate an ion gradient, which can stimulate the remaining neural circuitry of the retina or your bipolar and ganglion cells. So what does that look like? So hopefully everybody in the audience has a nice healthy retina, and you're sitting in your office right now, and what's happening is the light from that office is hitting your retina, and it's being picked up by molecules in your photoreceptor cells that's being sent to by your bipolar cells, ganglion cells, and then to the optic nerve. If you have retinitis pigmentosa or macular degeneration, what happens is you lose those light sensing cells. You lose your rods and cones. And so as those cells start to degenerate, your eyes become incapable of taking light and converting that light into a signal that can be sent to the brain. So what we do is we put our light activated implant where those native photoreceptor cells would exist. Now the protein is going to absorb light. In response to light, the protein is going to pump protons, which will be picked up by receptors on your bipolar and ganglion cells, and then that will transmit a signal to the optic nerve. Now, there are some competitors out there, but most of these competitors are very hardware-driven technologies. And so what you'll see is these are engineering marvels. But with that added engineering and complexity comes a much more complex surgery, where we're looking at over 4 hours for Second Sight's technology, over 3 hours for Pixium's technology. Then you have to deal with the external hardware. And then what's most important here is that they really still offer very low digitalized resolution. Because we are not limited by the number of electrodes that can be placed on a chip, but rather the molecular packing of protein molecules on a scaffold, we get a much greater pixel density, which allows us to have a much higher resolution than these competing technologies. Again, because we're using a light activated protein, we don't need external hardware to activate the implant. So to give you a sense of what that increased resolution looks like, we have Dr. Burge on our left-hand side. We have the current standard of care in the middle with that very low digitalized resolution. And then this is what 260 pixels per millimeter squared would look like on your far right. Now, there are a number of emerging technologies that are under development. Optogenetics is one of them, which I like to think of as sort of a fancy word for using light-activated proteins to restore neural function. Some of the challenges that optogenetics is facing currently are the types of proteins that they're using. So they're either using these channel rhodopsins, which are potassium pumps, which absorb in the ultraviolet range, or they're using these halorhodopsins, which are chloride ion pumps, which absorb more towards 600 nanometers. Bacteriorhodopsin absorbs at 560 nanometers, so we get the whole visible range, so we don't need any goggles or special effects to be able to absorb incident light. Gene therapies, for retinitis pigmentosa, what I didn't mention is that it's controlled by over 100 different genes. So, if you have 100,000 people in the US, 100 different genes, that's about 1,000 patients per gene. So it's very difficult to treat those patients globally. And so what we're doing is the technology that we're developing, it doesn't matter the gene type that you have. So we're able to treat all of those patients with retinitis pigmentosa. And then with stem cells, the challenge that you have there is that it's very difficult to recreate the entire retina and get it to all of those cells to talk to each other in the appropriate way to get a functional vision or a visual percept. Additionally, one of the challenges that optogenetics has, and I'm going to get into this with how we actually manufacture the retinal implant, is that optogenetics is often one of the biggest challenges is the type of light intensity that it requires to stimulate enough, to generate a sufficient gradient to stimulate those neural cells. And so one way that we get around that is actually how we generate the artificial retina, which is by creating multiple layers of this light activated protein on the scaffold so that we can absorb a sufficient incident light in order to generate that gradient. And so this is what that layer by layer process looks like. So right now we're doing this in our laboratories in Farmington, Connecticut, right across the street from Dr. Lee. And we're doing this using an automated dipping apparatus where we alternate layers of our protein, bacteriorhodopsin, and then a polycation, and then we deposit this onto the ion permeable membrane. Now we treat the bacteriorhodopsin so that it's consistently oriented so that all the protons are going to be pumping in the same direction, which is really important in order to get this gradient to stimulate those cells. At the conclusion of the manufacturing cycle, we resize the implants, we package them, and then we terminally sterilize them. And so what you'll see in this image on the right-hand side, there is a relationship between optical density and the number of layers. The more layers you add, the greater the optical density, the greater the signal. Now, the challenge with doing this is that every time that we add one of those layers, there's the potential for irregular deposition. And so when we do this terrestrially, this process is very much subject to the effects of gravity. We have issues with sedimentation, which can reduce the implant homogeneity. We have issues such as evaporation, surface tension, and so all of that can lead to inefficient and irregular protein deposition, which ultimately leads to a decrease in the performance of the artificial retina. It also leads to a reduction in the usable area that we have available to us for preclinical experiments. So it makes the quality assurance and assays that are necessary to evaluate the protein-based artificial retina require much more rigor. So how do we get involved in space at all? In 2016, we were part of Mass Challenge, and this was very, very serendipitous. We were actually sitting at a table, and the participants from CASES came over, and they said, "We're having a panel presentation. Anybody who's interested, we're partners here, and we're looking to provide funding for people who can use microgravity to benefit people on Earth." And so, I went to the meeting, and I knew that we had a challenge in how we manufactured these artificial retinas. Microgravity was not out of the question, but I had no idea how to do research on board the ISS. And so when we initially thought about this particular project, we were trying to automate the whole process ourselves. And little did I know that you guys have lots of implementation partners and a lot of people there that can actually help with this process. So over the course of our about 12 to 15 weeks at Mass Challenge, this project evolved. We eventually won the Mass Challenge Cases Boeing Prize at the end of 2016, which allowed us to conduct our very first experiment on board the ISS, which was to look at the effects of microgravity on the layer by layer deposition process. So we flew on SpaceX 16 at the end of 2018 with the support of our implementation partner, Space Tango. And what you'll see here on your left-hand side is our actual cube lab. The center image shows the cube lab with the cover off. So you can see there's lots of tubes and wires and pumps that are allowing the fluid to pass over our scaffold and the protein to pass over our scaffold to get the deposition. And then what you'll see on the right-hand side is you'll actually see the purple color of the protein in that middle chamber actually flowing over the scaffold. So what we were able to demonstrate is that we were actually able to successfully layer on the scaffold. You'll notice in that last image there on the far right that we did get some bubble formation. So that's something that we are working towards improving for our future flight, which is anticipated in Q1 of 2021. But overall, what's important as you're thinking about this ISS National Lab experiment, we're looking at improved homogeneity, stability, and then performance of the overall implant. This is for us, it's very practical because the protein is very, very stable, as I mentioned very early on. So we can do manufacturing in an ambient environment, so we don't need a lot of cooling or heating. This process is entirely automated, so we're limiting astronaut time as well as resources on board the ISS. And then, as this is looking towards the future for other technologies, thinking about commercialization on the ISS, this is a very, very feasible and manageable for us as we're targeting the orphan indication of retinitis pigmentosa first. So our production volumes in the supply chain are going to be a little bit lower or more manageable in the beginning. And then we can think about scale as we move towards macular degeneration. One important thing that I think is worth noting that we learned from our SpaceX 16 flight is that the protein functioned the same pre and post flight, which is something that I know a lot of people who are working on board the ISS, you use microgravity to look at different protein functions. But what you'll see pre and post flight here is that the protein maintains its function and ability to pump protons on a relatively similar time scale, which was really important for us. So some of the key learnings from this first flight, we were able to demonstrate layering in microgravity. We were able to deposit 100 layers onto specific areas of the substrate. As I mentioned, we did get some bubble formation, which we have now been able to eliminate, but that was a challenge in that very first flight. We didn't see double layering. So in this terrestrial process, what we need to do is make sure that we can only coat one side of the implant, because we want all of those ions to pump in one direction. If you get coating on both sides, you have pumping in both directions. So the way that we were able to layer this onto the chamber allowed us for uni coating on one surface. We did see a little bit of cross-contaminations of the solutions, which we're working to mitigate for this future flight in 2021. This work allowed us, you see a little tab at the bottom, we were able to get a phase 1 SBIR following this, where we're looking at the optimization of parameters for this for future flights. This also led to us getting a phase 2 SBIR, which we have now, which is actually looking at exploring what we've learned from these first couple of flights to other technologies beyond the artificial retina and applying this layer by layer process to other technologies, which I'll talk about in the next couple of slides. But what's really important here is, this year we also were able to secure, along with Space Tango and NASA NRA. And the goals of this NRA award were to explore the benefits of microgravity to produce our artificial retina on board the ISS. And so what we're doing here is evaluating these on-orbit manufacturing processes to produce artificial retinas that will then be examined back on Earth. And so a lot of what we're doing right now is working with regulatory people, along with some other officials at NASA, to establish good manufacturing processes that are necessary to produce these products on the ISS that will have clinical benefits. So it's definitely a learning experience for everyone involved. So in order to do this, our goal here is really to manufacture an artificial retina that's going to be safe and effective. We want to be able to demonstrate consistent and reproducible manufacturing processes. And then a lot of what the NRA is also supporting is assay and characterization development for this artificial retina, so that we can be able to examine the effects of microgravity on this deposition process. So what we hope as we begin to move forward with this project is starting to explore some of the other applications beyond the artificial retina where we can use layer by layer deposition. So as I mentioned, or as Ken mentioned a little bit in my background, what I've worked on during my PhD career is using bacteriodopsin for a number of different device architectures. So you'll see here the artificial retina, optical data storage, associated processing, pattern recognition, and then toxin detection. All of these technologies are still under development, and so can be used, and we can use layer by layer deposition to work on these technologies. And then beyond that, things like biosensors, wound healing, tissue engineering, biofouling are some other applications where I can see layer by layer being used to have clinical benefit moving forward. So with that, I would like to thank you for your time and look forward to some questions at the end.
Thank you, Nicole. Bacteriodopsin, 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 3 times.
Tongue twister, even I get stuck.
Thank you. That was a really great presentation. I'll tell you, both of these presentations for me, first of all, the research is terrific, and the technology innovation that goes along with just getting these things implemented and executed is amazing. But the other thing I would take note of here, I think these are great examples of what the construct and the public enterprise, public service of a national lab in collaboration with NASA can bring about. I would argue that without having that enterprise, that institution to sort of serve as the convener of, I think, otherwise disparate interests, and I'm talking about funding, engineering, research expertise, microgravity expertise, miniaturization, the whole 9 yards, without having the ability to pull those pieces together, I don't think these things come to light. So great presentations. Moving on to our next presenter, we're going to have Dr. William Wagner. Dr. Wagner is the director of the McGowan Institute for Regenerative Medicine, as well as the Distinguished Professor of Surgery bioengineering, and chemical engineering at the University of Pittsburgh. Dr. Wagner holds a Bachelor of Science from Johns Hopkins University and a PhD from the University of Texas in chemical engineering. He currently serves as chairman of the Tissue Engineering and Regenerative Medicine International Society, Americas. He is the deputy director of the National Science Foundation Engineering Research Center on Revolutionizing Metallic Biomaterials. And William is the chief scientific officer of the Armed Forces Institute of Regenerative Medicine. I can also say that Dr. Wagner is working in collaboration with CASIS and several other National Lab stakeholders and partners in academia and industry, and is really proven to be an effective expert and leader of a series of symposia focused on how we might leverage microgravity to further different interests in the area of regenerative medicine. So Dr. Wagner, welcome, and we would love to learn more about your team's focus and your interest in conducting microgravity research in the future.
Thanks, Ken. So this is going to be, as they say on Monty Python, something completely different. This is not going to be research focused per se, but more trying to answer the question, how does one effectively utilize low Earth orbit for biomedical production, for development and commercialization? And the approach I'm going to take is I'm going to talk about the institute that I direct and how we've gone about accomplishing our mission, which is fully dependent upon commercialization, and then how we're working with a large group of stakeholders to try and build a structure that would facilitate, that would catalyze commercialization in low Earth orbit. So just in terms of background of where I'm from, McGowan Institute for Regenerative Medicine, we're in Pittsburgh, and we're in Pittsburgh because of a fellow by the name of Thomas Starzl. And if you don't know Tom Starzl, there's a couple of books on him. He really was the pioneer of modern organ transplantation. And in the late 80s, he put Pittsburgh on the map as the place that was really doing the key innovation in immunosuppression that was allowing successful heart, liver, lung, other soft organ transplantation. So what Tom Starzl did is he made the situation that Pittsburgh was a place people from around the world went to if they needed a transplant. Since that time, many things have changed. Things are much more equitably distributed. There's rules for organ sharing. There's highly organized organizations that do this. But back then, it was more of a Wild West kind of situation. So what happened is in Pittsburgh, we had this patient volume of people waiting for organs. Even back then, there were not enough organs to go around. It's still the case. And that created industrial interest, because if you were developing an artificial heart or an artificial liver, lung, this was the most ethical patient group for you to test your technology, was in someone who was waiting for a transplant. So you would try to keep them alive. You'd bring them back from the brink of death by implanting this device and hope to keep them alive until the transplant would become available. That's called bridged transplantation, and that's still done today. And this was a paradigm that got set up in Pittsburgh, is that we had a lot of industry knocking on our door and doing trials with our clinicians. So what we recognized in the early 90s was we had the front row seat of watching organ support technology. And I can tell you as an engineer, we had a front row seat at seeing organ technology fail. And every good engineer knows that there's nothing like failure to provide instruction in terms of designing something better. So what we did is we brought in our engineering faculty. We actually embedded engineering faculty within our clinical departments, our graduate students and basic scientists to understand what was happening in organ support technology and to develop the next generation. So that led to the formation of our institute. This is the mission statement. I'll give you a short mission statement. Our mission is to develop technologies that address organ and tissue failure. That's the elevator pitch version of what we do. This is a longer version, and you can see we educate, we train, we generate scientific knowledge, and we develop and deliver therapies. But this last one is critical. because a corollary to our mission that I always say is if we do not get our technology to patients, we have not accomplished our mission. We have fallen short of our mission. And for us to get our technology to patients, we have to commercialize, and we have to be cognizant of what the commercialization ecosystem is like. So that means many things in the biomedical space. You heard some of them from Nicole right before. Regulatory pathway, intellectual property landscape, good manufacturing practice, good laboratory practice, business model, competition. You know, all these kinds of things we're trying to integrate further upstream in the process, so that if I've got this really cool idea that I want to get married to about how I'm going to make the next heart support technology, and it's so complicated that it'll never get manufactured, I want to know that earlier so I don't spend 3 years making great scientific publications, but at the end of the day not having anything. So this philosophy of integrating commercialization thought processes earlier is a key part of what we're trying to do. So we've done this with a number of different effort areas, with a number of different partners, and I want to highlight just a couple of them. One is with the military. So after 9/11, when Operation Iraqi Freedom and Enduring Freedom were ramping up, and the terrible IED results in the warfighters were coming back into Landstuhl and to the U.S., it became apparent to us that regenerative medicine might offer some hope to these patients, and ultimately, if we were successful, we might be able to get some clinical trials going with regenerative approaches. So we partnered with Wake Forest University and about 25 other universities around the country. We formed this consortium that was funded by the DoD. It actually is in the end of its second phase. It's still going on right now. And we did over 21 different clinical trials, filed over 100 different patent applications. And these covered areas ranging from burn to muscle loss to complex tissue replacement, a lot of craniofacial related reconstruction. So I think the military would say by any measure they considered it to be a success. And what we learned in working with the military, especially with the frontline docs that were dealing with these injuries, is the patience was short. They did not want to wait for a long time for some theoretical benefit. They needed something soon, and they needed something they could treat their patients with. So we had a very strong emphasis in getting to clinical trials. Another current consortium that we have going on, we're really right in the beginning of a third 5-year phase of this, is Regenerative Medicine Resource Center for Craniofacial Technologies. It's in partnership with University of Michigan and the Weiss Institute at Harvard. And this came from the National Institute of Dental and Craniofacial Research at the NIH. And their observation was they had funded a lot of what's called R01 research, meaning their basic research grant, but they hadn't seen a lot of it getting to patients. There was some intellectual property generation, but things went off into the so-called valley of death and never got to the patient. So they stood up actually they stood up 2 consortia. We're one of them, there's another one in California. And what we do is we basically shepherd, we mentor ideas from academics, from small companies through this multi-gate process to get them towards phase 1 clinical trial. That's our objective, is get them to that phase 1 clinical trial. So helping them with regulatory considerations, interactions with the FDA, manufacturing, and bringing in the resources that they need and the evaluations, the business model evaluations that they need to get investment and to move forward. So you can see right now we've got 19 different funded programs, everything from TMJ replacement to dental bone interface replacement to even things that are more simple in terms of preventing dental caries. So this is a project that, again, is very focused on that commercialization. The third one is funded by the FDA, and this one is in the neglected population of pediatrics and pediatric devices. What many don't appreciate is a lot of devices are never tested specifically for pediatric populations. And what is done is a device approved for an adult is simply downsized or applied as best seen appropriate by the physician at the time. So the problem is there's not big economic drivers because the pediatric market tends to be small. So what we're doing with, again, with support from the FDA is we've partnered with Children's Hospital of Philadelphia and formed this Pennsylvania Pediatric Device Consortium. We're, again, one of several within the country. And we focus on underserved needs, from very complicated devices like pulmonary support for pediatrics to fairly simple mechanical devices that allow intubation to happen more readily in a small patient. And again, we're applying GLP, GMP, business modeling, intellectual property support to get them to the point where they can get the resources to get to the patient. So what does this have to do with microgravity and low Earth orbit and the International Space Station? Is what if we could organize a consortium, fund a consortium that would do the same type of resource accumulation and making available these resources to shepherd concepts through the commercialization pathway in low Earth orbit. So it would be something like this very simplified stage gate process where you have defined gates, you know, in the development process. These would all be broken down with much greater detail. There may be a regulatory first interaction with the FDA, device designation by the FDA, proof of concept in vitro, in small animal, et cetera. And basically defining what resources are needed to help those meritorious projects move forward and to judge ourselves by how effective we are in moving things through this pathway. So we put together this concept in coordination with ISS National Laboratory of these three phases. The first one was to conduct, which is what we're doing now, an online series of thought leader workshops. So assemble thought leaders and define value propositions. Why would anyone want to spend their money to do something in low Earth orbit to accomplish this? What's the return on investment? Why couldn't they do it terrestrially? You know, basically the fundamentals of the value proposition. Like I say, that elevator pitch, why you're going to do it. You just heard a beautiful example from Nicole. You got it. You understood why you wanted to do it in space. So doing that with all these different other technologies, these candidates. Then proposing this public-private alliance structure and soliciting and applying funds to de-risk, to validate and mature technologies towards commercialization and clinical translation. And then finally, implementing and managing that and moving things, literally moving things through that that pipeline. So we're in the middle of our workshops right now. We've done about 15. We're probably not quite to the middle of it, but they've really been terrific. We've had a lot of good participation. Our objectives are, as I said, assemble these thought leaders, and we've pre-identified three promising areas: stem cell production in microgravity, organoids or microphysiological systems, so-called organs on a chip, and leveraging their behavior in microgravity, and biofabrication technologies, which Lambda Vision would definitely fall into that category, as would 3D printing and other technologies. And then also we're bringing in robotics and artificial intelligence expertise. We had a beautiful talk this morning from one of IBM's experts on artificial intelligence in chemistry and biology, and how can we marry that into supporting this translation. Secondly, as I said, we really want to focus on value propositions. What are the gaps in our knowledge that we could fill with resources that would effectively de-risk the technology? What fundamental understandings? Could we get a business consortium around organs on a chip and they would say, yes, we want to know how this happens in microgravity and we're willing to fund part of that. Quantitative business models, robust business models, not qualitative that we often see when we write our proposals, but things that you have put in front of an investor that would not elicit anything but a serious consideration of that investment. And then industrial consortium, as I mentioned, getting consortium members to come together and identify investments that they would find valuable. Then finally, we want to prioritize the findings of all these workshops that we're in the middle of right now, summarize the current status of what's happened to this point from a technological development targets, and then formally forming a national consortium that would be funded by stakeholders, private sector, public sector stakeholders. So to wrap up here, as I said, we're in this multi-phase symposium right now. It's three steps: an educate phase where we basically took people who knew robotics or knew what was going on the space station, and people who knew regenerative medicine, and tried to get everyone up to the same speed in terms of understanding and focusing on the most advanced technologies. And now we're in this generate phase where we have these intense 1-hour sessions where we really try to dig down on a given topic and identify what's the promise, who would the investor be, what would the business model be, and importantly, what is blocking that from happening right now. And then finally, come end of the year, December, January, we will prioritize. These are what we believe to be the most important points for investment, and then put together different versions of that white paper for marketing, as I say, to the stakeholders to help stand up this consortium and begin moving things forward. So right now we're using Microsoft Teams. This is just an example of the liveliness of this. I had to turn off my notifications for this talk because the one from this morning was still pinging me. But a lot of input as we do this asynchronously as well, talking about what we think the different challenges are. So I'm very happy about the amount of engagement we've gotten across the stakeholders. So I will stop there and turn back over to you, Ken.
Thank you, Bill. That was awesome. As you said, it was I think a completely sort of different perspective than what maybe the other two panelists were talking about. But also, as you mentioned, as you referenced Lambda Vision, and even I think there's some applications to what you were talking about to Sage-In Lee's work. And what I really enjoy seeing here is we're starting to sort of see this more seamless integration of what we're doing here on Earth in laboratories and in developing businesses, and looking at other factors to consider in program development other than just the qualitative, scientific, and technological merit of things. And so it's great to see that evolving and getting into our ecosystem. So thank you. All right, let's get into some discussion and some Q&A with the audience. I think we have a few questions. The first one is for you, Dr. Lee. This comes from our ASGSR audience. The question is, does the overexpression of myostatin affect the number or percentage of circulating white blood cells?
I don't have any information about that. So we haven't noticed anything, but we haven't done a very systematic analysis to address that specific question.
Okay. We'll see if we can get back to the audience participant, but right now we don't have that data. I wonder, Alex, I wonder if you have any questions for any of the panelists based on what you heard today.
Yeah, thanks, Ken. Thanks to everyone for just great presentations. Really exciting to see the results, and exciting to see the kind of formation of new alliances out there, kind of just emerging organically from the opportunities that are available. The question actually for Nicole, certainly have been watching the development of the artificial retina. Certainly, it's something that the NASA administrator has spoken about a lot. Certainly, lots of folks are very hopeful that something may emerge there, because of course what you're talking about is a type of in-space production. And I guess what I wanted to get a sense from you is, what are your projected timelines, right? There's a lot of stuff that still has to happen before this becomes a product that is regularly manufactured and part of essentially the service of ameliorating macular degeneration. What timeline do we think we're looking at for a clinical trial with something that was made in space that is then being being actually be used, and then what kind of timeline would we be looking at for something that could be actually a market-based service?
Yeah, that's a great question. I think, a lot, you'll hear this over and over again, I think, as you're thinking about manufacturing and especially some of the timelines of matching the timelines of industry also to doing what we're doing on board the ISS. Um, but we are targeting to have our retinal implant into patients within the next 2 to 3 years. A lot of the timeline here, again, is going to be a little bit limited by the types of studies that the FDA is going to want us to do, both preclinically as well as what it's going to take to manufacture this artificial retina as well. Because this is so just to the FDA is going to be looking at this as a drug, because the primary mode of action is going to be the bacterial rhodopsin. But we are going to be doing much longer animal studies because this is something that's going to be put into the eye and it's going to stay in the eye for the duration of the patient. Um, so a lot of these studies are going to be long-term studies. But we're estimating within 2 to 3 years for the clinic, and then hopefully after that, so that brings us to 2023, 2024, another 2 years till we can start to market this for the orphan indication of retinitis pigmentosa.
Well, that's great, and I'll just say that at least that encourages me because that means that is within the timeline of what we hope the ISS will be available for. So, I know you're working as hard as you can, and as always, any feedback appreciated, and anything that the U.S. government can do to help.
We certainly are, moving quickly with this project. We just were on board NG14 with our protein to do some evaluation, and we're targeting our next protein manufacturing flight on NG15 in the beginning of 2021. So we are working really quickly, and we're working with a lot of different, consultants to help us kind of put together as much GMP process as possible before that next flight, which is going to be, I think, a critical proof of concept for us.
Yep, look forward to it.
Great. Dr. Wagner, question for you now. So based on your experience at the McGowan with translation of biomedical technology to clinical application, and taking into account at least what you've learned thus far in some of the workshops that you've been participating in, are there any, I don't know, red flags, markers, capabilities, things that we might need to facilitate and catalyze the industrialization of biomanufacturing in space? Anything that you've flagged early on that we need to be thinking about now?
Well, it may not make me popular with astronauts, but I think non-human based platform future, robotic platform future, where a sterile environment, for instance, could be maintained, cells, processes, everything would be done robotically and with remote sensing. I think that that allows specialization and the level of control that one might need for the production that the FDA would want to see. But I'm optimistic from everything that I've learned over the past several years in terms of the likely growth of low Earth orbit platforms from the private sector, stepping up, and the private sector will continue to step up as costs go down, and that'll open up more business models. So I think the future with robotics, again, marrying small scale robotics, sensing, as much of this mechanized as possible, I think that's an important step.
That's great. All right, thank you. All right, we have another question from our audience. This is for Dr. Lee. So Dr. Lee, is there a way to conduct mice experiments without human intervention on the ISS? If so, can you maybe give an example of how they might do it?
I think that might be a little bit above my pay grade, but I would say that it'd be hard for me to imagine doing a study that would not involve any human, intervention at all. But certainly there are studies that can be done where that could be minimized. But even so, the mice have to be cared for, so the cages have to be cleaned out and all of that. So until that technology, gets to a point where you can avoid having any human involvement at all, I think having astronauts on board, skilled and motivated, is going to be critical to this.
Yeah. I would throw out a question. Maybe it's slightly related, perhaps not. I'm sure most folks have heard about Commercial Crew Program. We're going to have additional crew flying the space station in the very, very near future. Got a launch coming up, and that's going to expand our in-orbit crew time tremendously. We're essentially going to almost double up the amount of crew time that astronauts, crew members can devote towards research and utilization. I wonder if Nicole, you were there doing research on station, you as well, Sejin. I wonder if you might think for a moment. It seems like, what can we do on orbit? What gaps do we have in capabilities as far as either near or real term observation, analysis, analytics, and rapid iteration in orbit, as opposed to having to wait for a launch, so that we can better leverage the crew time? What are maybe some of the tools that you were missing when you went and did your experiment, that you would like to have on station that you have at your lab, that would allow for example, iterative science that would take advantage of a crew time? Nicole, have you thought about that, or did you observe anything?
For our very first flight, because we're trying to make this so automated and be able to, a lot of what we're looking at from a manufacturing perspective is a lot of quality control, which we can get, through imaging and cameras. So we haven't really encountered much of that as a hurdle. However, as we are starting to move towards looking at GMP, good manufacturing practices, and starting to put some of those processes and procedures in place, I think having a good set of historical data, a good set of, whether it be temperature, pressures, those types of things that give us a sense of environments, those are going to be important moving forward. I think a lot of times, when companies are looking at putting something forward for FDA approval, they rely on the fact that some other groups have used certain animals historically, or that there's, they can point to literature precedent. A lot of that is stuff that we're starting to look at now. What precedent is there that we can leverage, and what things do we actually, what pieces of data do we need to collect in addition to that, in order to manufacture the implant with the rigor that's necessary for FDA approval.
Very good. Sejin, any comments on that you were might want to weigh in on?
Yeah, I'm not sure that this is an answer directly to your question, but I think certainly the challenges of doing mouse studies, aboard the ISS, are huge. And what I mean by that is, first of all, there's a limitation in terms of the number of animals, that can be housed up there and taken care of. And that also relates to the amount of astronaut time that's required for even basic procedures. And so for a study like this that we would carry out here on Earth, we would have almost certainly had other experimental arms to the project. We would have probably increased the number of replicates to make sure that we get, achieve statistical significance. There would have probably been additional assessments done along the way, but everything that gets done on Earth gets magnified by, an order of magnitude in terms of the time required up at the ISS. And given the time constraints, on the astronauts, we had to design the study in a way that we probably would not have done on Earth. But that's just the reality of doing studies up there.
That's great. That's great. Alex, did you have any follow-ups for any of the panelists?
No, I guess, just as kind of a last question there, at least for me, we're obviously, looking at a future that, at some point will include a platform in low Earth orbit beyond the ISS, right? And it's an exciting opportunity. I personally think we're still a ways away from that. Legislation is certainly going through with dates for either 2028 or 2030, depending on whether you're looking at the House or the Senate bills for the ISS extension. But at some point, we do need to think about a new platform. And we know that these platforms take a long time to develop. So, and I understood the point that in an ideal world, maybe there's a world where you have uncrewed platforms. That's not really NASA's mandate. NASA's mandate is to continue to do its crew work. So we're going to be continuing to fund crewed platforms. So, thinking about some limitations on that, if we think about building a new platform, what new capabilities, right, would you all want to see on a new platform that may, be fielded, say, in a decade's time, right? If you could change something around, if there's something new that you thought was coming on, what's something that we should be looking at for a new permanently crewed platform in low Earth orbit?
I think you've stumped the panelists, Alex.
Good. I didn't know that.
You've given them carte blanche and mum's the word. I think we heard from Dr. Wagner, certainly robotics and AI is something that seems like it's going to be pretty critical, at least for some of the biomanufacturing applications. And I think in many ways, both cases and NASA, especially NASA, have started to go down that path. There's no doubt about that. Seijin, any comments on that?
I think I would just follow up on what I was saying before, which is that I think at least for the life sciences, for I think the microgravity really provides a unique opportunity to look at effects in a whole animal that's really not easy to duplicate here on Earth. It really is a unique opportunity. But in order to do those types of studies, I think increasing the capacity in terms of the number of mice that could be, in a study at any given time, I think would be of enormous help.
That's great. Bill, did you have a thought?
Yeah, I would say I think there needs to be movement towards having more fundamental resources for biological experiments on station, and less having like an organ on a chip going up and then being studied, and instead assembling the organ on a chip on station. And that helps get to, on the in vitro level, it helps to address the issue of number of observations. So being able to have fundamental structures, having the cells there, having an astronaut, seed them and assemble them, ultimately I think it needs to be robotic, but that would allow greater throughput, thinking about how do we store these cell tissue resources on station.
And Nicole, last word on this question?
Yeah, I mean, I think I would kind of echo what Bill said as well. I mean, I think as we start to see more fundamental research on board the ISS, and we start to address many of the questions that he mentioned during his presentation, I think we'll start to figure out where the gaps are a little bit more. I think, if we start to build this consortium, we can start to identify a lot of the key players and key stakeholders that will allow us to put different pieces of equipment on board, whether it be, some type of a station to allow for more rigor. And, I guess also to Dr. Lee's point, something that allows for scale. If there is a way, I think we're getting really good with the automation. But, for something like we're doing, is it now we can just transport protein, and that can be, then it's assembled up there. I mean, what does scale actually look like, and how do we actually start to get to that point? Those are some of the questions that I think are hopefully going to be addressed in the next couple of years.
Well, that's great. Yes, me too. Great panel. I think we're approaching the end of our time. And so we'll just wrap up. I'd like to first and foremost extend a very gracious thank you and appreciation to all of our panelists, Dr. Alex McDonald from NASA, Dr. Nicole Wagner, Lambda Vision, Dr. William Wagner, McGowan Institute, and certainly Dr. Seijin Lee from Jackson Laboratory. Thank you. I think your presentations were awesome. As much as we have been celebrating and acknowledging 20 years of continuous human habitation of the ISS and what has been accomplished over those 20 years, how much we've learned about living and working in space, exploring space, then moving into research utilization phase, and learning how do we leverage microgravity and leverage the environment of space as a value proposition to perhaps move industrial technologies and applied R&D from that awesome mulch of fundamental science that we learn from and those learning cycles that have occurred over the past couple of decades. I can tell you, hearing all of these presentations, it's really exciting for me to think about what the next 20 years are going to be like. And not only on this platform as we grow and evolve off of the current platform, which is the ISS, onto other assets in LEO and even other orbits in space. Hearing all of this, I can tell you it's quite exciting, and it's never been a better time. As I mentioned, commercial crew and some of the other things that we're doing, seamless integration of what we do on Earth and space, just fantastic. So best of luck to everyone. Please stay in touch with the National Lab. We're going to have some amazing opportunities in this next year and following years, and it's going to be a fantastic next 2 decades. Everyone, thank you and have a great weekend. Take care.
Thank